Hello everyone and welcome to the NPTEL course in Pharmacognosy and Phytochemistry. In this week, we are going to focus on drugs which have their genesis from primary metabolites, and those are carbohydrate based drugs. In your biochemistry, we have already studied what are carbohydrates, but let's visit some basic concepts to understand these drugs better. So, carbohydrates are often referred to as saccharides. They are the source of energy for plants and animals. In plants, they are stored in form of starch, whereas in animals you will find them in form of glycogen. If you see them structurally, they are referred to polyhydroxy, aldehydes or ketone derivatives, chiefly consisting of carbon, hydrogen and oxygen, and that's the reason they are called as carbohydrates with the formula CnH2nOn. That means if you just take a simple example of this molecule called as alpha D glucose, where N is equal to 6, H2N means your twice 6, that is 12, and O6. So, most of the carbohydrates go by this formula. Say for example, alpha and beta in this formula, the alpha refers to the anomeric hydroxyl group. So, the hydroxyl group on anomeric carbon, if it is below the plane, you call it as alpha, otherwise it is called as a beta hydroxy if it is above the plane. Now, this carbohydrates have been found abundantly in plants and are products of photosynthesis. So, from photosynthesis you get your water and carbon dioxide producing glucose, and from glucose you have variety of pathways which lead to diverse sugars. Some of the sugars polymerize by forming ether linkages, and then they form what are called as polysaccharides. So, if it is 1, it is referred to as monosaccharides. If it is 2, you can refer it as disaccharides, trisaccharides, and upwards are called as polysaccharides. Now, animals are not photosynthetic. We don't carry out photosynthesis, but we have a ability or a unique pathway called as gluconeogenesis, in which glucose or sugars can be synthesized from proteins or lipids. So, they form a source of sugar in case if we fast or if we have a diet which is lacking carbohydrate, our body will convert proteins and lipids into carbohydrates, because carbohydrates are primarily the molecules which are used to generate energy. Now, if you go to see the physical and chemical properties of carbohydrates, if you have a single monomeric, what we call it as a monosaccharide group, take for example glucose that we just saw, monosaccharides generally occur in crystalline free flowing form. If you consume them, they are sweet to taste and they are easily soluble in water. So, simple examples include your glucose, fructose and so on. Now, when two such monosaccharides join by ether linkages, now this linkage is depending again on the nature of the hydroxy group. If this hydroxy group happens to be below the plane, we call it as alpha glycosidic linkage. If this group happens to be above the plane, you call it as your beta group. So, below the plane you have alpha group, above the plane you have it as your beta group. So, depending upon that, the sugars or saccharides join to each other either using alpha and beta linkages. Now, why are they linkages important? If you see your saliva, they contain numerous enzymes, and one enzyme is alpha amylase. So, take for example your starch. Now, starch, the glucose molecules are linked by alpha linkages. So, an enzyme called as alpha amylase can hydrolyze it, and hence your starch can be converted into glucose. But say for example, when you eat cellulose, something like a rope is made up of cellulose, or your cotton is made up of cellulose. Now when you consume cellulose, in cellulose if you see here, the linkages are beta linkages. We have alpha amylase. So beta linkages cannot be hydrolyzed by us, and as a result cellulose will remain undigested in our gut. So just a difference in the linkages changes the digestibility of the saccharides. Now this can go on adding more sugars to form polysaccharides. When they form polysaccharides, they are no longer crystalline. So disaccharides are crystalline, sweet, soluble, and they can easily break down to give you energy. Say for example, a disaccharide can be like a sucrose, which will hydrolyze to give you a glucose, and fructose, both of which will give you energy. But take for example polysaccharides. Now this is a polymeric group. You will require a lot of enzymes to hydrolyze it, and hence the digestion of polysaccharides is very slow. Hydration and solubility of polysaccharide is a time-taking phenomenon. So they take time to hydrate, they take time to swell, and more time to dissolve. So they are very resistant to dissolve in water, you can say. Now depending upon what sugars are linked, if the same sugar is linked as a polymer, say for example same glucose is linked as a polymer, you call it as homosaccharide. But if a different sugar, say for example one is glucose, one is fructose, one is arabinose, or different sugars are linked to each other, in that case you call them as heteropolysaccharide. Examples of homopolysaccharide include starch and cellulose, whereas examples of heteropolysaccharide includes your agar and pectin. Now what are sources of carbohydrates? Carbohydrates are primary metabolites. They are found in most of the living organisms, but when you think of carbohydrate drugs, let me give you few examples. Plants are the chief sources of carbohydrate drugs. So when you say your acacia, dink when you call it, tragacanth, pectin, starch, inulin, all of them come in from your plant sources. Microbial sources, you get substances such as xanthan, gellan, villan, gum, and so on. From marine sources, you get carbohydrates which are transparent jelly like your alginates, carrageenans, which is used in your toothpaste, agar, which is used in your microbial media. From animals, you can get your carbohydrates such as honey, which is a product of your bees. So once you have or break your honeycomb, you can get an abundant saccharide, and that is also an instant source of energy for us. Now coming to carbohydrates, carbohydrates are further derivatized to form a set of complex molecules, and these complex molecules can be divided or can be classified or can be called as gums and mucilages. Now what are gums and mucilages? Let us try to understand. Gums are molecules which are generally or polymers or polysaccharides as you call it. Both of them are polysaccharides, but gums are polysaccharides that are generally an outcome of an injury given to a plant. So we can say that are pathological product. So when you hit the plant, or when you cut the plant, or when you give injury to a plant, the plant tries to synthesize something to counteract the pain, and that process is called as gummosis. In this process, it will generate a gum to close the wound and to heal itself. Whereas mucilages is something you can see in your chia seeds, or you can see in your flaxseed. When you're consuming it, or even in your okra, that is your lady finger, when you cut it, there is some slimy thing coming out of it. Those are naturally present, and physiologically those polysaccharides which are present are called as mucilages. So gums are pathological, they are generally protective in nature and found as a response to damage, whereas mucilages are more physiological, they are there to act as a reserve food or stored food. Whereas if you see gums, they will easily dissolve in water, they might have initial period which is required to hydrate. Mucilages swell, They clearly do not dissolve, the reason being they are located inside the cell. So when you see those chia seeds in your sharbat for example, you will see that the extra layer remains on top of seed and does not disperse or dissolve in water. The reason is it is located inside the membrane and that doesn't allow it to flow freely. Examples of gums include acacia, tragaca and karaya gum, whereas mucilages, you can see that jelly thing which is there inside the aloe vera leaf or isabgol or even your flax seed or chia seeds. Those are all examples of mucilages. In terms of chemical composition also there lies some difference. Now when we saw polysaccharides, we said that these are sugar molecules which are attached to each other. Now imagine the sugars getting converted into acids and we call them as uronic acid. So say for example glucose will get converted into glucuronic acid. So they are slightly acidic as compared to your polysaccharides. So gums are polymers of sugars and sugar uronic acids, whereas if you see mucilages, they contain sugar uronic acids and some of them form esters with your sulfate molecules. So you have, in addition to your uronic acid, also sulfate molecules being present that makes them more clear and more comparatively transparent as compared to your gums. Now let's start with a gum containing drug, and the most simple example I can give you is acacia. Acacia is an Indian gum often referred to as gum arabic or gum acacia or simply acacia. If you see the biological source in terms of USP as a reference, it is actually obtained from Acacia senegal, which is a plant which is more native to East African countries, mostly Sudan and the Senegal region. That's where it is harvested from the wildly grown plants which are a thorny trees, whereas in India the plant which is more cultivated for this is your Acacia arabica. Both of them yield good quality gum and belong to the family Leguminosae. Few other places from where you can obtain your gum is Morocco and also some African countries. But if you see in terms of supply, majority of the acacia supply still comes from Sudan. So you can see here, once the injury is done, the plant starts exudating and this exudates form a tear like appearance and the tears are collected. So let's see the processing. So what is done is this plant, generally from wild or sometimes it might be cultivated, is allowed to grow. You can take it from a young plant but the gum quality is not so good. So ideally it is allowed to grow for a period of 6 to 8 years, and within the season, generally the dry season as they say from November to February, what is done is a small incisions are made in the bark. Sometimes the bark is removed. Now here you have to be little careful while removing the bark that you do not expose the xylem. The reason for this is if you expose the xylem, then because it's a good supply, the ants and other insects find their way out. Once the ants and other insects find their way out, the tree doesn't yield gum. So even if in some cases during the cutting, if the xylem is exposed, it's covered again so that the ants and other insects don't infest it and the gum exudation continues. So cuts are being made and the gum starts oozing out. Now it takes time, the process of gummosis takes time, and a good seasonal variation is seen in the purpose. So if you do it in the summer season, the process of gummosis is fast. The reason they say is the microbes are much active, the infection happens fast, and as a result to counteract the infection, the trees produce the gum quicker. So in the summer season you will get the gum within 3 to 4 weeks. In winter season the infection runs very slow, and as a result your gum will be produced in a much longer duration, that is 6 to 8 weeks time. Once the gum is produced, it is kind of scraped off from the tree and dried in the sun. During the drying process, all the moisture which is present in the gum is kind of evaporated, and from the translucent or clear tears, as we saw, the gum becomes more and more hard and opaque. So it becomes brittle, it can be crushed, and on crushing it appears like a cream colored powder. So you can get acacia as tears in the market, or you can get it as crushed white powder, which is slightly buff or brownish in color. It has absolutely no odor. It has a bland, a little mucilaginous carbohydrate, but not a sweet taste. And you might even come across small, tiny tears in varying concentrations or varying dimensions. It is insoluble in alcohol, being a saccharide or being a carbohydrate derivative, but is soluble in water. Now, what is it made up of? If you see gum arabic or acacia, it is said to be made up of arabin. Now, what is arabin? Arabin is basically salts. You can say calcium, magnesium, or even potassium salts of arabic acid. And arabic acid is a polymer which is generally made up of galactose, rhamnose, or arabinose. So, you will get that upon hydrolysis, but the chief backbone of it you will see is made up of D-galactopyranose, usually joined by 1,3 linkages. Now, where do you use acacia? Acacia in India is generally used to make laddus, but pharmaceutically it has a great utility. It is used as demulcent, it's used as a viscositive, and because it enhances the viscosity, it is used as a suspending agent. Because of its carbohydrate polar nature, it is used as emulsifying, binding agent, as well as stabilizer. Now, these polymers have also found utilizations or uses in novel drug delivery system, in formation of micro encapsulation. So, you can encapsulate a powder or a drug or a oil using gum arabic. It is also used in spray drying powders. It is used in tablets. So, during the process of tableting, initially the blend is to be made, that is a dough, and that dough is converted into granules. So, this is used for the process of granulation. It is very compatible with other carbohydrates and proteins, and hence it is a good excipient, a good compatible excipient. It is used to make candies, other food products. Now, because of its viscosity imparting property, it is used as an adhesive like a glue, a binding agent for marbling colors. It's used in ink, in cosmetics. Acacia also goes in making of your eyeliners and mascara. Now, going to another carbohydrate based drug, and that's your agar. Now, this agar comes from marine sources. It's also referred to as agar-agar. For those who are from food industries, it's also referred to as Japanese isinglass or vegetable gelatin, vegan option. So, it is called vegetable gelatin because it gives the same jelly like consistency, transparent jelly like consistency, but is obtained from plant sources, basically algal sources. Now, what are these algal sources? You can obtain agar from numerous species. So, you can obtain it from Geladium amansi. Now, this is something which is a algal species or a red algal species which is cultivated in Japan. Initially, the art and craft of making agar was exclusive to Japan, but with its widespread uses, people started cultivating this red algae throughout, and other algae were also explored. So, in Australia, if you go to see, the agar comes from your Gracilaria species. If you see New Zealand, it comes from Pterocladia species and other allied species. Other countries which also produce agar include your Korea, South Africa, US, Chile, Spain, Portugal. So, most of this who have a proximity to sea have started exploring this red algae and starting gaining agar from it. So, how do you produce it? Preparation of agar or cultivation of agar has been done in Japan. I'll show you, this is the shallow sea where the sea depth is not greater than a meter. So, what is done is bamboos are kept with string and the sea kelps are planted. They are allowed to grow on the strings for a period of time, and during the summer season they are taken out. Now once they are taken out, they are dried. They are dried and beaten for the reason because you will see some shells or some sand will start infesting and growing. Some sea creatures or marine creatures, barnacle like creatures will start growing on it, and we don't want it. So they are shaken so that this extraneous debris which has accumulated on the sea kelp falls off. Now once that is done, you can still get rid of it by washing it thoroughly. Now this is washed with water so that all the other dirt, grime and other debris is removed out, and then this can be easily bleached by keeping it in sunlight. It is seen the pigments of this red algae are sensitive to sunlight, so the moment you expose it, a discoloration happens. So they are periodically rotated over a period of time in sun, and water is sprinkled continuously so that the pigment discolored. Now once that is done, you have a good clean a red algae. Now this red algae is taken in a tank, and about 50 times its weight of the algae, you will take the water. Now this water generally is acidified water, and it is acidified either with acetic acid or with sulfuric acid. So you to break the cell walls and you to take the algal mass out of it. So it's pulverized, it's boiled well, and the mass is dissolved. Now once it is dissolved, you will see that it forms a viscous solution. Now this clear viscous solution is filtered when hot, because on cooling it will tend to congeal and form a slimy mass which is difficult to filter. So as it is hot, it is filtered and then allowed to set in trays. Now once it is allowed to set in trays, what happens is you get the blocks, like a tray block of it. This is again finely ground with help of knife or coarse ground. Then what is done is the surface area is increased. This is again kept for drying. So every time it is ground and again kept for drying, you will get a size reduction and dehydration. So more and more water loss happens. Now in this case, if you take a particularly climatic advantage of Japan, what happens is when you take this or set it in tray and kind of grate it or grind it, during the night when the temperature is cold, the water within the sea kelp crystallizes to ice, and this ice later on during the daytime melts to form water and evaporates. So this freeze thaw process for water continues, and eventually you get a good grade algae. This can further be dried to get a fine product which can be dried as sheets, which can be dried as threads, or which can be granulated to form powder. Now we can do it industrially as well. On a commercial scale, you need to take this sea kelp same way, get rid of all the dirt, debris and barnacles by washing it for a day or so, keeping it exposed to water, and then heating it extensively with the help of steam heated digester and exposing it to acid, where it will hydrolyze and form a jelly. Now this jelly is subjected to deep freezing and it's made or converted into ice, and what happens is this ice are then broken, heated, exposed, evaporated, and put into a rotary vacuum filter, so that whatever moisture content it is will all vanish. So you get a nice powdered agar which is then used in microbial media. So you can see here, either once you do it, you will get a powder like this which is a granulated agar, or sometimes it is extruded to form a string like appearance. So this is how it is. So agar is yellowish or little yellow white to grey. It has a mucilaginous taste and may occur as strips, flakes, sheets or coarse powder. Now when they are in form of strips, you generally get a big, like 60 centimeter long strips with just a small 4 millimeter diameter or width. When it is form of sheets, you get about a 50 centimeter big sheet with a 10 centimeter width. Now if you take agar in cold water, it doesn't dissolve. But if you boil it in hot water, it forms a clear jelly. So 1% will form a clear jelly and then it will set it. And that's what you use as your microbial media. So few chemical analysis of it will tell you that agarose is made up of two polymers, the linear one being agarose and the branch one being agaropectin. Now agarose is more of a neutral polymer which is responsible for the gel or a clear gel properties, which is made up of D and L galactose unit, whereas agaropectin is mucilaginous, or what we call it as a sulfonated polysaccharide, which will yield galactose plus more of uronic acid derivatives which have been esterified with sulfuric acid. Now agaropectin is something which is responsible for viscosity of agar solution. You can evaluate the genuine agar by few tests because it's mucilaginous and it contains sulfate. If you take this agar, hydrolyze it by HCl and then you treat it with barium chloride. Barium chloride is a water soluble salt, but it will react with the sulfates of agar to form barium sulfate. Now barium sulfate is water insoluble, so barium sulfate precipitates down. So if you get a turbidity, a white color powder being formed which settles down, understand it's your genuine agar. Same way, because of the sulfates, there is a dye called as ruthenium red which will bind to the sulfates and it will convert the mucilaginous matter into red color, which is not going after washing or diluting with water. You will see chunks which are red in color, and that test will tell you that definitely there are sulfates and your drug is mucilaginous. You can also do the gelling test. Just take a 1% solution, heat it profusely to form a clear jelly and then cool it. If it sets, it's agar. And one more thing you can do is you can check it with iodine. Starch tends to form blue color with iodine, but if it's agar, it will give you a crimson red color. So this also distinguishes it from acacia as well as tragacanth. Now uses, agar is chiefly used or extensively used in microbial medias. It is used as laxative because we cannot digest it. It's a suspending agent, emulsifier, gelating agent for suppositories. It's a surgical lubricant. It's also used as tablet excipient, especially a disintegrating agent and an encapsulating agent like we saw in acacia. It is used in ointments and in some cases as a polymer in dental mold for an impression base. It is used as nutrient media for the reason being your bacteria cannot consume or digest agar, and that's where it doesn't lose its consistency and it remains stiff throughout. It is initially used in making photographic plates, but now it is discontinued. It is used for making clear jellies in cosmetic industries, and along with alginates it might be used as a thickening agent in food, especially confectioneries and dairies where you want those clear jelly like consistency but you don't want to use gelatin. It is also used in sizing preparations for silk, paper, in dyeing, in printing industries. It is used for printing fabrics and textiles, and because of its adhesive properties it is also used in or as a glue. So here's to few more references if you want to read more on this topic, and thank you everyone for your patient listening.